Okay. So good morning all of you. Welcome to all my beloved students in these lectures. Myself Parthasarathi Pandit, lecturer Food Processing Technology Department from Sheikhpara A.R.M Polytechnic. Previously, I have already discussed about the definition of milk, composition of milk, factor affecting the composition of milk, transport, handling, and grading of the milk, also testing of the milks, different testing. Today, we will discuss very important things, that is the microbiology of milk. When we're talking the microbiology, your mind always drawing a picture, that is the microbiology means bacteria, yeast and molds. All you are known little bit or elaborately from your microbiology subject. I will just focus only the microbiology of milk, only milk. Okay. Now, bacteria are microscopic, all you are known, unicellular fungi, which occurs principally in the form of spherical, cylindrical, or spiral cells, and which responds and which reproduce the transverse fissions in milk, and it produces the spherical and cylindrical forms are predominant. Most bacteria vary from 1 to 5 microns in size, and molds are multicellular, differing gently in most respects from bacteria. Although the individual cells are not visible at the naked eye, at maturity they may be observed readily as mycelium. They are found in soil, feeds, manure, and poorly washed utensils. And yeast are unicellular. When when a question arises, what is the difference between molds and yeast? That means yeast are multicellular and yeast is unicellular, are somewhat large in size than bacteria. Spores of yeast are readily destroyed during pasteurization process. Okay, and all you are known about the type of microbes, that is there is three types: psychometric, mesophilic, and thermophilic. All you are known. So when when the microbes grow the temperatures from ranges 5 to 7 degree temperatures, this is called the psychophilic. Psychotropic microbes, when can grow at temperatures ranging from 20 degree to 40 degree centigrade temperature, that is ambient temperature, is called mesophilic. And when can grow at temperature above 50 degree centigrade temperature is called thermophilic bacteria or thermophilic microbes. Okay, so microbiology analysis, why needed? Why we need to analyze the microbes? We are all known when microbes are produced in milks, maybe get infected. When we consume this full of microbes with milk, our human body can affect it. So this microbiology called pathogenic microbes, all you will know. Now, when we concerned about the milk and public health, that is how to how to microbes can grow rapidly in milk. There is the several steps. That is the when inspection of milk directly from the cow. We are sometimes in our daily life we taken milk, okay, but we don't know the milk, that is the milk actually milk taken from cow. So if cow is infection or cow is infected any disease, and this milk we are consumed, then our body also affected. Okay, or direct contamination of milk by human beings, indirect contamination of milk by human beings, and infection from man to cow and then to milk. So that is the factors where milk can be infected and our body also infected. Okay, so now focus on the my slide, that is the market milk control and grading, improving the milk production environment and practices, screening of milk supplies to access their stability for processing of manufacturing of products. And now we are all known, that is the several microbes, that is the yeast, bacteria, molds, also sometimes spores, E. coli forming. E. coli is the main microbes present in milk. And all you know the unit of microbes, that is the CFU or colony forming units. Now we'll discuss about the various techniques where we can easily count this type of microbes. There are several techniques, that is the various microbiology tests performed in dairy industry. When you are going to in any industry, in dairy industry, you will see there are lots of testing done there. So DMS or direct enumeration of total bacterial count, or direct microscopic count estimation of number of viable bacteria cells, that is the standard plate count or SPC, assessing the microbial metabolic activities, that is the dairy reaction test, detection of specific contamination, that is the coliform or pathogens, estimation of biochemical changes of metabolics form in dairy products as a result of microbial growth, that is the activity, gas production, toxin production, etc. Now we will discuss about the method, that is the DMC or direct microscopic count. The DMC method enables rapid enumeration of bacteria cells along with their study of morphology of the total bacterial count in milk and cream with minimum equipment. It consists of examination of stained film of a measured volume of milk or milk products spread over 1 centimeter square per area and dried on a glass slides under microscope. Some somatic cells, shapes and arrangements of bacterial cells present in film can be easily and rapidly visualized and recorded. Now DMC, when calculated, DMC per ml equals to n into mf, where n equals to average number of cells per field, mf equals to microscopic factors, and mf equals to area of simmer by area of microscopic into 1 per volume of milk, that's equals to 10,000 per 3.1416 into r square. This technique is very useful for screening of milk supplies on the receiving platform of a dairy plant, as well as for grading of milk. Now SPC or standard plate count, all you are known about that, because in our food industry always will, when we involved to count the microbes in different food samples, we have to done the SPC methods, that is the standard plate count methods. In these methods, a known quantity of milk sample is diluted to known degree and equal portion of each dilution is poured into the Petri plate, followed by the addition of nutrient agar medium. A technique known as pour plate methods, the medium is allowed to solidify after mixing the content by gentle rotation of the plate. The organisms present in the sample are expected to be trapped in the agar gel. The plate are subsequently incubated at 37 degrees centigrade temperatures for 40 to 72 hours. In principle, each organism is expected to take up a separate position in the medium and grow into a mass of cell of a size sufficient enough to be counted by naked eyes. Recognized as a colony forming unit, as earlier I have said, that is the unit of the microbes, CFU. Hence, a colony count performed at this stage represent number of viable bacteria present in this given volume of sample. The major limitation, basically this is a drawback of this method, that is the some bacteria which is not produced in these agars or not trapped by these methods, we cannot count it. Okay, so this is the drawback of these SPC methods. Now, MBRT or methylene blue dye reduction test, this is a very important test for milk industry. When you look any industry, any dairy industry, anywhere, all perform this test because this is the most important test. By this test, we can understand basically the extension or shelf life of the milk. Okay, now there are certain dyes, lot of dyes here, but we used only methylene blue dye because methylene blue has a oxidation and reduction properties. Bacteria consume dissolved oxygen during the growth of milk and consequently reduced the OH to a level at which these dyes are reduces. Basically, when methylene blue has two properties, oxidation and reduction. So when oxidation, dissolved oxygen present there, and microbes get this oxygen and easily grow up. And when it reduces, it becomes dye becomes colorless. So we can assume, so we can show that the test is over. Okay, such dyes can be employed to assess the biochemical activity of bacteria and thus estimate number of bacteria indirectly. So now look forward that the test is when at least this test already done in your lab. Take 10 ml sample in a sterilized test tube, add 1 ml of MBRT dye, close the tube with sterile rubber stopper and it stand in a water bath, temperature maintained 37 plus minus 1 degree centigrade temperatures, and note the time taken from decolorations. So results, when the milk get colorless after 30 minutes or 2 hours, we can say that this is the poor quality of milk. And when we observe the milk is colorless after 8 hours, we can say that the milk was excellent quality. Same time and same methods, that is the research-driven reduction rate test. This is the almost similar to the MBRT test. This is also a dye. So also in the indicators to reduce the liable reduce the bacteria, reduce the blue dye taken place in two stages. First, the dye irreversibly reduces to a resorphin undergo through a series of colors changing from blue to pink. During second stage, resorphin is reversibly reduced to a colorless compound dihydroresorphin. Okay, so it is almost similar to the MBRT test. Now, coliform count, it is the very important because coliform is the main pathogenic bacteria in our human body, and also it get affected human health. So every industry have to count to coliform, and obviously all industry maintain the FSA guideline rule or PFA guideline rules where they prescribe the limitation of the count. Okay, so the coliform group of bacteria, that is the Escherichia coli interbacters, includes gram-negative, non-spore forming, aerobic and facultative rods capable of fermenting lactose into the lactic acids and gas. As per American Public Health Association or AFA methods, coliform is milk are detected by following scheme. That is preservative coliform test. 1 ml of milk sample or decimal dilution is poured to sterile plate, followed by the addition of 10 to 15 ml of violet red bilagars or VRBA. The content of plates is by gently rotating and tightening each dish, and finally agar is allowed to solidify and incubated at 320 degrees temperatures for 24 hours. Appearance of typically red color colonies measuring about 0.5 mm is taken as positive test. Alternative fermentation tubes of 22% of brilliant green lactose bile broth are incubated or inoculated with the sample, and production of gas after incubation of 48 hours at 320 degrees temperatures is considered as positive indication of presence of coliform test. So your exams is always a common question, that is the how to calculate or how to indicate or how to assume or how to identify the coliform in your milk sample. Another method is the done by inoculation of meconkimbab broth with 0.1 ml of milk samples, or examined for the production of acid detected by changing the color of the medium from purple to yellow. This is also a test for identify the coliform. Now, yeast and mold count. Potato dextrose agars is the medium of choice for yeast and molds count, as potato extract promotes the growth of these organisms. The suppression of bacterial growth, which may otherwise interface, is ensured by adjusting the pH of medium by adding 10% tartaric acids. The plate count technique is used, and plate are incubated at 0 degrees in temperatures for 2 to 5 days. So, thank you all of you. Please follow the our council website, because all the notes already uploaded here. Please download it and read it. Okay, so thank you.